Good morning everyone. We'll again begin with the biology class of today, that is the plant physiology. We'll start with the plant physiology part of the sections as given in the NCERT textbook. In this, the plant, hope you have just gone through the retained part and the deleted portions of the chapters. So already if you just go through that in the plant physiology, the two chapters, that is the transporting plants and mineral nutritions, are being deleted. Only few part is being retained, while there are in plant physiology there are 5 chapters, that is the transporting plants, mineral nutrition, photosynthesis in higher plants, respiration in higher plants, and plant growth and development. Yes, but in this, as per the present COVID situations, some portions are being retained while some portions are being deleted. So in that, the transporting plants and mineral nutrition mostly are being deleted, while the other chapters, that is the 13th and the 14th. 13th, photosynthesis in higher plants and respiration in higher plants, will be dealt in the later classes. Now we will go with the 15th chapter, that is the plant growth and development. So we will go with the in plant physiology with the 15th chapter, plant growth and development. So what do you understand by plant growth and development? Already we have learned about the cell, the types of cells, and as a living organisms, the property of a living organism is to respond to the stimulus, to grow. Yes, it can divide. Growth is by cell division. So let me give the introduction of the topic plant growth and development. All living organisms have this capacity of cell division. They divide and grow into a well-developed organism. If you take the example of a human beings only, the two gametes fuse together, form a zygote. The zygote develops to form an embryo. The embryo divides and divides and divides, redivides to form an adult organisms. Similarly, in the plants also, the growth starts with the germination of the seeds. The seeds germinate, producing the radicals and the plumule. The radicals will grow to form the root system, while the plumule grows to form the shoot system. So this overall growth is the growth of plants is also initiated by the process of cell division, and the process of cell division is being initiated by the chemicals produced in the plants, and those chemicals are nothing but the hormones. As you know about some of the hormones, like the hormones secreted by the pituitary gland, the thyroid gland, the pancreatic gland, and other glands. But plants don't have such glands. Though they don't have the glands, still the chemical substance, that is the hormones, are being produced. So overall, as thyroxine is helping in overall development of the cells in animals, in plants also such chemicals or hormones are being produced. They are being produced so that it can help in growth and development. So in simple definition, what is a growth? It is an irreversible change with increase in size and weight of an organism. Just it increases. It is an irreversible. It cannot be reversed. Growth is an irreversible process with increase in weight and change in the structure of an organism. So for this, the main substances are the hormones. Commonly we call them as plant growth regulators. We call such as plant growth regulators, in short PG. These hormones or the growth in plants is also specified by by the presence of the hormones. So these hormones, based on their functions or the activities, they are being classified into two forms as plant growth promoters and plant growth inhibitors. In plant growth regulators, they have been classified into two types: plant growth promoters or plant growth inhibitors. As the word says, promotes. These are the hormones which initiates the process of the growth, or which induces the cell to divide or the organism to grow. So these hormones, these are such chemical substances which are produced by the plants which initiates the growth. Which includes proper cell division, differentiation, cell formation, or formation of a particular organ like the roots, stem, or the leaves, including the flowers and the fruits. Overall, those chemical substances which are involved in the growth of an organism, of an organ in a plant, are called as plant growth promoters, which includes auxins, gibberellins, and cytokinins. These are the hormones produced by the plants which help in promoting the growth. And plant growth inhibitors. Inhibitors means which will slow down the process of growth but undergo the process of de-differentiation or differentiation, so that at that moment of time the activity of the cell is being stopped and it is modified to perform another functions. For example, the abscisic acid and ethylene. So in hormones, in the plants, some are promoters and some are inhibitors. Inhibitors, they slow down the process of the growth, which may help in the formation of fruits or flowering period. So we'll go in detail how these hormones were being discovered, who discovered these hormones, what was the fact for the discovery, or what are the physiological roles of these hormones. So first we will begin with the concept of auxins. What are the physiological role of auxins and who discovered the hormone auxins? The first hormone that we will be studying is about the auxins. The Greek word, the Greek meaning of auxins is to grow. Now these hormones that were being discovered were all in an accidental practices of the scientists in their work, during their work, or in the process of their work. Many scientists work with that, then propose the thesis regarding to the work. These all hormones that were discovered were by accidental process. Actually, some scientists were working on the canary grass. So while they were working, they observed a very specific phenomena of the light and the response of the light towards the plants, or the response of a plant towards the light. So in simple example, if I say that the plants also respond to the stimulus, that stimulus is the light. If you take the example, the movement of the sunflower towards the source of the sun, or the touch me plant, the touch When we touch it, it closes or it encloses the leaves, that is the mimosa pudica. So there are all responses. So here also, due to light also, the plants also respond. We call that phenomena as photoperiodism. Light, the response of a plant towards the light, towards the source of life, is known as photoperiodism. So here the scientist named Charles Darwin and his son Francis Darwin. And his son Francis Darwin were working on canary grass. Canary grass. They observed that the extreme tip of the plant, is known as coleoptile, or we can say the tip, the shoot tip or the extreme apex of the stem is having a group of cells or a mass of cells known as the coleoptile. When they observe that it is responded towards the source of light. With some simple diagram, let me say you what was the concept of their experiment. So, the tip of the grass is known as coleoptile. So, this tip, when it was exposed to light, in simple word, if you say that photoperiodism, that is the response of a plant towards the source of light. So in this diagram, let us understand the concept of what they want to mean to say that what is the concept of that light and the plant. So here the tip of the plant, the grass, the tip of the plant is known as the coleoptile, or the extreme apex of the shoot is known as coleoptile. When it was exposed to a source of light, it responded and showed the bending movement, or there was a curvature, or we can say that it was slightly bended towards the source of light. But when this was being cut, when the coleoptile was being cut, but it did not respond towards the source of light. In the third step, this is the first step, second step, and in the third step. In the third step, the coleoptile was being covered by a black cap. Yes, why was being covered by a black cap? When it was covered by a cap, it did not respond towards the source of light. But when the cap was being removed, the coleoptile again responded towards the source of light. So once again, I will try to explain it. In the first setup, as it was the grass, the apex was being exposed to the light, that is the coleoptile when exposed towards the light, it showed the curvature, or it bended, or it responded towards the source of light. But when this coleoptile was being removed, when it was removed, it did not show any response. Similarly, when it was capped, a black cap was being placed on the collectile, then also it did not respond towards the light. Then when the cap was being removed, then it showed the response by bending towards the source of light. Then they confirmed that the light, when falls on the collectile, stimulates some chemicals in the region of the shoot apex, which made the meristematic region, or the presence of that chemical made the part to respond to the stimulus. That chemical was later on named as auxins, and the meaning of auxins in Greek language is to grow. Yes, the meaning of the auxin in Greek language is grow. So they just initiated that a chemical is being secreted by the collectile, which made the plant to respond towards the source of light. Yes, many experiments were being done, but it was later on the scientist called as Wendt. Wendt, who isolated the auxin from the oat seeds. He isolated later on with many experiments done by the scientist. It was W.F. Wendt who isolated the auxin from the oat seedlings and named these chemical substances or the hormones as the auxins. Yes, and stated that no auxins, there is no growth in the plants, or we can say that the growth of the plant or the development of the meristematic region like apical meristems or lateral meristems, the growth of such regions is because of the auxins. So from then the people started to know about the chemical nature of the auxins. So here first initiated by Charles Darwin and Francis Darwin, who put forward the concept of the auxins with their experiments with the help of a collectile. Yes, now next we will go what are the physiological roles of these auxins, how these hormones are helpful, or what are their physiological roles in the plants. What are the physiological roles of the auxins? As earlier said, that auxins is derived from a word. That word meaning is to grow. So let us go in step by step in the form of points. What are the physiological roles of the auxins? So here we will there in the textbook it is given in the form of a paragraph. To make it easier or to make it clearly, I am giving the concept in the form of points. First is apical dominance. Usually in plants there are two forms of buds, apical buds and axillary buds. Yes, apical buds help in the growth which are present at the apex of the plant, and at the base of the apical buds there are axillary buds. In some plants, due to the presence of the apical bud, they does not allow the axillary buds to grow. So in some plants it was being observed that the plant is just known as apical dominance. So these apical buds did not allow or inhibited the axillary buds to grow. So only the plants were just growing with tall, inhibiting the growth of the axillary buds. But when these auxins were being treated, yes, even the phenomena where apical buds dominate over the axillary bud is known as apical dominance. But when auxins were being added, yes, not only this suppressed the activity of the apical buds and help in the growth of axillary buds, where it started to produce many lateral branches on the stem and produced a bushy-like appearance. So here the auxins are initiated in apical dominance. So apical dominance is being inhibited by the auxins. Second is cell division and cell enlargement. So here the auxins induces the cell division and cell enlargement. So if you take the meristematic region, the meristematic regions are those regions which keeps on dividing, forming the new cells. So when the plants is producing particular organ like the radicals or the plumule, or we can take as the apical buds or the apical meristems or the lateral meristems or intercalary meristems. So these all meristematic tissues, if you just take the germination of a seed, the seed initially grows with a small radical, but as it grows, the cotyledon producing the primary roots and the secondary roots, where the primary roots grows producing the lateral roots. So this all growth from a small radical to form a shoot or for a root system. There is a growth of shoot and the root system is because of the auxins. So overall the auxins induces the cell division and the cell enlargement. Already we have learnt in the regions of the roots also. Yes, the tip region, the meristematic region, the elongation region and the maturation region, as you have studied in the topic of morphology. So these all regions, that is the growth of such parts, the regions of the growth. So here those all are being initiated by the auxins, which are responsible for cell division and cell enlargement, especially in the region of elongation, where the root grows, where there is the increase in the length, and in cell division, that is the meristematic region, they activate, these auxins activates the dividing cells and makes the cells to divide very rapidly. So their auxins are inducing cell division and cell enlargement. Then third one, it also induces xylem element differentiation. What are xylems? Xylems are water conducting tissues of the plants, which are made up of four components: xylem fibers, xylem vessels, tracheids and xylem parenchyma. So as the plant, the transport should be quick and fast for the process of translocation. So here this hormone, the auxin, initiates particularly the xylem elements. The xylem elements, that is, in that the two important translocating cells are tracheids and the vessels. So as the plant is growing, it initiates these xylem elements to differentiate so that there can be fast development which will help in the transportation of the substances. Why? If there is low concentration of auxins, if there is low concentration of the auxins in the plants, it will initiate and promote development of normal roots. In some cases or in some particular plants, when you add low concentration of the auxins to the plants, it will initiate. It is emphasizing or it initiates or promotes the development of the normal roots. The normal roots can be the primary roots, yes, and the secondary roots, especially in those plants where roots are being used or vegetatively propagated or which are being commercially used for the purpose. Yes, so low concentration of the auxins will initiate and promote the development of normal roots, while higher concentration of auxins initiates development of lateral roots, in the development of the lateral roots. So sometimes in some plants, if auxins are being treated with higher concentration of the auxins, it will initiate the development of the lateral roots. So these are the 5 points. Later on plants will go with the points. So apical dominance, cell division and cell enlargement induces xylem element differentiation. Low concentration will initiate the growth of normal roots, while higher concentration increases the development of lateral roots. Then, what are the other roots? Sometimes in some certain group of plants where there are some auxins that were being known like indole acetic acid, indole butyric acid and naphthalene acetic acid. So in some cases when application of such auxins, that is some of the auxins like applications of indole acetic acid, yes, some of the plants when they are being applied, the applications of indole acetic acids will lead to early rootings of the plants. For example, sugarcane and rose, or we can say reproduction is the cutting process or the stem cutting process, where the stem itself is used, the culm itself is used as a seed for the vegetative process. So at the cut tip ends of the sugarcane or the roots or the rose plants, if they are applied with the auxins, we can find there it will initiates the early rootings of such plants. So in such plants like sugarcane and the rose, when they are the cuttings, the cut ends of such plants, when they are applied with indole acetic acid, it will lead to early rootings. While the synthetic auxins like indole butyric acid and naphthalene acetic acid, these are both natural auxins. These are natural auxins which on applied on the plants, yes, which induces, it will prevent or it will delay premature fall off leaves, flowers and fruits. Or we can say that it prevents abscission of the organs. So the synthetic auxins like indole butyric acid, naphthalene acetic acid will prevent the premature fall of the leaves. That is, before they could ripen or before their development. If they fall, yes, if they fall, that is called as premature fall. But auxins will prevent the premature fall of the developing leaves or the flowers and the fruits. Or simply we can say that they prevent the abscission of that particular leaves or the flowers and the fruits. And it also induces in this, for example, we can go to the example here. That example here is pear and apples. To such plants, if these auxins are being treated, the flowers or the leaves, the fruits, they develop properly, they grow up to their level, they get ripened and produces the flowers and the fruits. So sometimes if they are not applied, they can be premature fall of the flowers of the pear and the apples. Then the next point is it induces development of parthenocarpic fruits. It induces the development of parthenocarpic fruits. Parthenocarpic fruits, those fruits, or in simple words if you say seedless fruits. The natural seedless fruits are the bananas, pineapples and the grapes, which are of natural. The seeds, they don't have the seeds. Yes, so such development of fruits where the ovules do not undergo fertilizations, then there is no formation of the seeds, or the plants, the natural parthenocarpic fruits which do not have the seeds or the seedless fruits are the banana, pineapple and the grapes. Those don't have the seeds. But application of auxins can also help in the development of such seedless fruits. Take the example of papaya nowadays. The papayas are not having much seeds in the present cultivation process. Yes, there are even certain plants also. For example, the tomato, brinjal, watermelon, and the papaya. When these plants are being treated with the auxins, then there is no formation of the seeds, or very less we can develop seedless fruits. Those are tomato, brinjal, watermelon and the papaya. Then the last point is eradication of weeds. Example 2,4-D. Eradication of weeds. Meaning of weeds is what? Unwanted plants. Now, if you take in a cultivation process also, the most weed that is being grown along with the crop plants is the grass. So, when you sprinkle such chemicals, it will not allow the weeds to grow with no effect on the crop plants. So weedicides, as there are insecticides to kill the insects, there are weedicides. So in that, one of the weedicide that will prevent the growth of the weeds is 2,4-D, 2,4-dichlorophenoxyacetic acid. So this 2,4-D is a widely used weedicide. Or if you take the cricket pitch, the pitch or the batting pitch of the cricket ground, where it is completely bare without the growth of the grass, while the whole ground is made up of grass. Or we can say that these chemicals are being sprayed in that particular part, so that there is no growth of such weeds. So these are all the physiological role of the auxins. So we had been in this class, that is in plant growth and development, how these auxins were being secreted and what are their physiological roles. So they were discovered by Charles Darwin and Francis Darwin, who initiated their experiments with the help of the canary glass, which showed the response towards the light, and later on what this chemical, how it will affect the growth in plants was being studied, and these all the physiological rules: apical dominance, cell division and cell enlargement, xylem element differentiations. What happens when applied low concentration? That means it will produce in the formation of normal roots. High concentration, it helps in the formation of the lateral roots. While some of the auxins, particularly the application of indole acetic acid, will lead to the early rootings or the stem cuttings. When applied to the stem cuttings, you can find the early rootings, for example, the sugar cane and in the rose. And the natural auxins with are indole butyric acid and naphthalene acetic acid, which will prevent the abscission of the leaves, particularly in pears and the apples. While it also induces the development of seedless fruits. Those nowadays we are getting some of the vegetables and the fruits without the seeds. That is because of the chemical auxins. Then eradication of the weeds. Here the weeds, the unwanted plants can be eradicated or they can be stopped. The growth of such plants can be stopped by the application of 2,4-D, which is widely known as the way. So in the next class, we'll go with the next hormone, the gibberellins, the cytokinins and their physiological role, how they were discovered. So here, who discovered auxins? and what are their physiological roles are the two important concepts of this class. So tomorrow we will continue with the same concept of gibberellins, cytokinins and the abscisic acid and the ethylene. Thank you.